the metabolism of the proteins and reuse of the amino acids affect what we're looking at?
So let me reinterpret the question. So what if we have a lot of proteins that were made radioactive, and then through time they get degraded, and then those radioactive amino acids get reused into a new thing, right? That's where the chase comes in. You're providing such a wallop of non-radioactive amino acids that they dilute out any possible effect of that sort. So essentially the answer is no. So everybody's all good? All right. So this is going to be an Instapoll question in just a second. Let me just set it up first. So the idea is that here we're looking at so let's just read it. Right? So we're going to have cells in cultures, all right, that we're going to be giving pulse and chase of different amounts of time, at which point we're going to break the cells open, and we're going to reach out with antibodies to do an immunoprecipitation of a particular protein. All right? So now here's the situation though. If I have my cells and I give them the pulse, and then I say, okay, give them a chase, 1 minute chase, and then I lyse them all, now I have nothing for my 5 minute chase. All right? So I have to actually set up multiple dishes, all of which are going to be pulse all at the same time. All right? And then all of them are going to start the chase at the same time. And then this dish, I'm going to immediately, right after that, I'm going to lyse open, break the cells open. So now, as soon as you break the cells open, you stop everything in the cell, all right? Not the metabolism doesn't continue there, if you do it correctly. So then I'm going to wait 30 seconds. 30 seconds later, I'm going to take my dish number 2, take the cells, lyse them open to stop the process, all right? And then 1 hour, that's hours or minutes, I don't know. I forgot, but whatever. It's hours. So half an hour, 1 minute, 1 hour, 3 hours, 6 hours, and 24 hours. All right. So I have multiple. Each of those lanes represents a parallel dish that was used in this experiment. Is that clear? All right. So we give the cells a pulse, 5-minute pulse of radioactive methionine. All right. You can choose what radioactive amino acid you use. You then wash the excess radioactivity and give them a chase of non-radioactive methionine, so that you swamp out any lingering leftovers of radioactivity in there, and now the proteins that are made in the chase are not radioactive. The only proteins that are made radioactive are in the 5-minute window of the pulse. So you incubate the cells, the dishes, for different chase times, at which time then you take the cells, lyse them open, and then you immunoprecipitate with an antibody against a protein called cathepsin A. Anybody knows what cathepsin A is? No surprise. You will know later. You run the samples on an SDS gel, and then expose the cell, the gel to X-rays to detect radioactive proteins. Okay, so here's a little variation, all right? In this experiment that I show you here, we had run the gel for a Western blot. We transferred the proteins to a membrane, then we tested the proteins with the antibodies, and then we exposed the film. That is not what we're doing here, all right? It's not a Western blot. In this case, you make your immunoprecipitates with the antibodies in the little beads, all right? You wash them, you boil that with SDS, that's what you run in the gel. Then you dry your gel, and then if we're hoping that there's radioactivity in the gel, if you put a Geiger counter, tick, 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 right in there, invisible. So you put a piece of film, and the film will be now exposed by the radioactivity and give you the image of the presence of radioactivity in the gel. That's what we're doing here. So this is why I say this is an X-ray film, and the only thing that is visible, there's lots of proteins in there, but the only things that are visible in the film are the radioactive proteins, and those are the ones we are interested in. Is that clear? All right. So that's what we do. So the predicted size from the gene bank information, we know that cathepsin A from the amino acid sequence should be 54 kilodaltons. All right. So this is the X-ray film of the experiment. This is at time 0, right at the end of the pulse. And then this is half an hour after the pulse, so 30 minutes of chase, 1 hour of chase time, 3, 6, and 24 hours of chase time. All right. So first question, which you all have in your handout, is right there. And let me set up the Instapoll. I'm going to go ahead and set up the next question. Don't answer it yet. Okay. So, which of these answers do you think is the right answer? Think about it and give me your best shot. I'm going to close it in 5, 4.
Are we just doing B? Sorry.
I'm sorry?
Just B or C as well? Just the first question.
Beers, yes.
Oh,
All right.
so we're just doing this
So
question.
just this one, just this one. Yeah, you don't have, we're not doing the other one. We're just doing this one. All right? So from what I can see here, most of you have one answer, so let's go through them. So cells make a lot of cathepsin A initially and just a little at later times. Why is that not a good answer? I think it's a good thing to analyze, not a strong message for suggestion for exams. When you're looking at the multiple choice part of our exams, it's important that you not only select your best guess, but that you really have reasons of why the other choices are not correct. Because if, very commonly I have seen through time, students go for distractors that are very well written and appear to be real but are not for important reasons. So you need to be able to say for each of the other answers, why is this answer not appropriate? Why is this answer not appropriate? So that you avoid mistakes during the exam. All right? So let's do that in here. So most of you say the answer is B. So, but let's analyze the other answer. So cells make a lot of cathepsin initially, but just a little at later times. Why is that not appropriate?
Because we're not looking at just the production of cathepsin, we're looking at the radioactive tag.
Okay. So, and that means.
It's also because it was pulsed all at the same time, so they were all made at the same time.
And exactly right. So the proteins that are, because we're only looking at radioactive proteins, the radioactive proteins were only made during that 5-minute window of time. We're not looking at what was made later, that all those proteins are invisible to us. So maybe, yes, maybe the cells made less later. We are not aware of that because those proteins are invisible. We're only looking at those that were made in that window of time. Even though we're here at 6 hours, we're looking at the proteins that were made at